Hi students, today we will discuss for the mechanism of biological nitrogen fixation. And this video it is in continuation with the previous video of nitrogen cycle. In the previous video we have discussed for the various steps which are present in the nitrogen cycle like ammonification, nitrification and denitrification. We have also seen that the various type of prokaryotes they are responsible for the biological nitrogen fixation. Some are free living, some are aerobes, anaerobes, and symbiotic association also present. So now we will continue for the biological nitrogen fixation in the symbiotic associations. And first step it is the nodule formation. Actually what we are discussing how the nitrogen it is converted into ammonia, right, through the symbiotic nitrogen fixing bacteria. In this diagram you will be able to see the root here and the bacteria. So bacteria they are present, right, and the roots of the legume they secrete the chemical attractants which are named as flavonoids, and you can name as betaines. These are what? These are the chemical attractants which are being secreted through the root hairs of the legumes, because we are discussing for the symbiotic nitrogen fixation here. So here the bacteria they are attracted, a hook is formed, and here the curling out of the root hairs it takes place by the release of nod factors. These nod factors they are released by the bacteria, and this is responsible for the curling out of the root hairs, right? And after that the infection thread it is formed, and this infection thread it reaches the cortical cells. This infection thread it reaches opposite to protoxylem points of vascular strand, and not only the infection thread it reaches, here the bacteria is multiplied as well. So now the cortical cells they are affected, and the infected cortical cells they become differentiated, and the nodule formation takes place. The swellings, nodules are what? The swellings. The infected cortical cells they differentiated and start dividing to produce nodule. Now here in the differentiated cortical cell after the nodule formation, the bacteria they stop dividing and they are present enclosing in the cell membrane, host cell membrane, and now the aggregation of the bacteria it is called as bacteroids. Please don't get confused here. First of all, the infection thread it is formed, and after that the cortical cells they are infected, the cells infected, and here the nodule formation takes place by the division of the cortical cells, and this nodule it is the swelling, and after the nodule formation here the infected cell, they become enlarged, bacteria stop dividing and forms the irregular polyhedral structures which are called as bacteroids. Where they are present? In the cells. And these bacteroids, they release a pinkish pigment called as leg hemoglobin. So this pigment actually, it is what? It is an oxygen scavenger. This pigment, it is formed by the polyhedral structures which are called as bacteroids in the cells. In the infected cells, they are nothing but the bacteria, right? Here the infected cell enlarge and bacteria stop dividing and form the irregular polyhedral structures called bacteroids, and they release the pigment which is called as leg hemoglobin, which is the oxygen scavenger. What it is having the function, you know, the nitrogenase enzyme which is responsible for the conversion of nitrogen into ammonia, it requires the anaerobic condition. And this pigment, which is like hemoglobin, it actually creates the anaerobic condition for the action of the nitrogenase enzyme, right? So, much important pigment it is. And here the enzyme nitrogenase, what it is a pigment having the iron and molybdenum. That is why it is called as the iron and molybdenum protein. It is containing the iron and molybdenum, and it take part in attachment of a molecule of nitrogen. So this attachment actually, you know, the the attachment leads to weakening of the bonds between the two nitrogen atoms. Here you can see the nitrogen atoms, they are having the bonds, and because of the iron and molybdenum present in the nitrogenase, it it they take part in the attachment of a molecule of nitrogen, and they are responsible for the weakening of the bonds between the two nitrogen atoms. And here the weakened molecule of nitrogen, it is acted upon by hydrogen from a reduced coenzyme, and now what is formed? The diamide is formed, right? Here after that hydrazine is formed. After that ammonia. This is what the motive, because if you recall your memory for the previous video, what the nitrogen fixation is, the conversion of nitrogen into ammonia, nitrate and nitrite. So here you have seen how the ammonia formation takes place, right? Uh, and here the enzyme nitrogenase, it works, and here the proteins which are present, like this is a protein which is containing the iron and molybdenum, and here the ammonia formation takes place through these steps, right? Now the reaction you can write. How you can write the reaction? Like this. This is what the reaction is. (16 ADP and 16 inorganic phosphates). And you can see much energy it is required, right, for the nitrogen fixation. So this is the story of the symbiotic nitrogen fixation, like the mechanism. If you discuss for the free living nitrogen fixers, so free living nitrogen fixers like azotobacter, they act upon the dead and decaying matter, organic matter, like the excreta, dead proteins, etc. And after that, the ammonia formation takes place, and the process is called as ammonification. This is for what? This is for free living nitrogen fixation. So ammonification, where the bacteria, putrefying bacteria, they act on the dead and decaying organic matter, and the ammonia formation takes place. And you can write here the reaction: protein, amino acid, and organic acid. And in this case, the ammonia is also released. Right? Now, you can also write here for the nitrification. Right now, what we are discussing? We are discussing for the free living nitrogen fixers, or you can say for the free living nitrogen fixation. So here the first, the nitrite formation takes place. Ammonia, it gets converted into nitrite, and after that this nitrite, it gets converted into the nitrates. And the bacterias already we have discussed for that. Here in this conversion you will write for the nitrosococcus and nitrosomonas, and in this reaction you will write for the bacteria nitrobacter and nitrocystis. Process nitrification. Next, denitrification, where the conversion denitrification, where the anaerobic condition, here the microorganisms, they convert the nitrate into the simple nitrogenous compounds and nitrogen. And the bacteria, they are like Micrococcus denitrificans, Thiobacillus denitrificans, and Pseudomonas denitrificans. You can write here the reaction. Right? Again, ammonification, where the protein, it is converted into ammonia. Nitrification, where the ammonia is converted into nitrate. First the nitrite is formed, then the nitrate is formed. And the denitrification, where the nitrates, they get converted into the simpler nitrogenous compounds and the nitrogen. And the bacteria, they are also different here. Right? Now let us discuss for the nitrate assimilation in plants. This is point of concern here. Right? This involves the reduction of nitrate to nitrite. So first the nitrate, it gets converted into nitrite. We are talking about the nitrate assimilation in the plants. The nitrates absorbed by the plant roots, they get converted to amino acids and amides before incorporating into proteins and other macromolecules. And the reduction of nitrate into ammonia, it is called as nitrogen assimilation. So here first step, when the nitrate, it is converted into nitrite, and then the nitrite, it gets converted into ammonia. This we can write as like NO3, right? And here you can write for ammonia. And here the enzyme, that is nitrate reductase. And here the enzyme nitrite reductase. Here you can write as FAD FMN, flavin adenine dinucleotide, flavin mononucleotide. These are the coenzymes here. And here you can write for ferredoxin. This is iron sulfur containing protein, and it is an electron carrier actually, right? So here again you have seen in the nitrate assimilation, the ammonia formation takes place. Now this ammonia which is being converted here, or which is you have seen here also in this nitrogen fixation, how it is used, right? And this is used in the synthesis of amino acids. Right? So first ammonia at the physiological pH, it gets protonated, and after that the amino acids, they are formed, and you can write for two processes here: reductive amination, and you can write for transamination. Actually the ammonia, it is not used as such, you know, it has to be converted into amino acid. Like first through the ammonia, from the ammonia the amino acids, they are formed, and now the amino acids, they get grouped, polypeptide is formed, which is the protein. So the ammonia formation we have seen in the assimilation, and here the synthesis of amino acid, it is the next step, where the ammonia, it gets protonated at the first at the physiological pH, and after that you will be able to see here the reductive amination. What the reductive amination is: the ammonia which is formed by the nitrogen assimilation, it reacts with the alpha keto glutaric acid, and what is formed? The glutamic acid is formed. Transamination, where the transfer of amino group of amino acid, it takes place to the keto group of keto acid, right? So here the transfer of amino group of amino acid, it takes place with the keto group of keto acid. And here the enzyme, it is required which is called as transaminase or amino transferase. So here you can write glutamate or glutamic acid plus oxaloacetic acid, and what is formed? Alpha keto glutaric acid plus aspartic acid. And in both the reactions, the enzymes you can write here, like here the glutamate dehydrogenase. And here the amino transferase or transaminase. Right. So this is what the transamination. Transamination is what? Where the it involves the transfer of amino group from one amino acid to the keto group of a keto acid. And the glutamic acid is the main amino acid from which the transfer of NH2 it takes place. Now enzymes, they are equally important, so be cautious for that. And now, you know, one more topic it is left, that is assimilation of ammonia. Assimilation of ammonia, it actually involves the formation of amides. It involves the formation of amides where? In higher plants. What are amides? They are the derivatives of amino acid in which OH component of carboxylic group it is replaced by the amino group, right? So they are the combination of ammonia and amino acids, and the two most common amides, they are glutamine and asparagine. And these are the components of proteins along with the amino acids. What the what the amides, they are having advantage? Amides perform the two main functions. One is the storage of excess nitrogen, and the second is transport. You know, amides contain more nitrogen than the amino acids because the additional NH2 group it is there. So they are transported to other parts of the plant via xylem vessels. And here I would like to add one more point, children, that in addition, along with the transpiration stream, the nodules of some plants like soybean, it export the fixed nitrogen as ureads, right? And ureads are what? They are having the high nitrogen to carbon ratio. So this is what the mechanism behind the nitrogen fixation. And I think we have discussed much for the nitrogen fixation. A quick recall if we want to take, children, nitrogen cycle where the circulation of nitrogen we can see, and in that the ammonification, the conversion of protein into ammonia, nitrification, the conversion of ammonia into nitrates and nitrites, and denitrification, the conversion of nitrate into the nitrogen. Here the symbiotic nitrogen fixation involves the invading of the bacteria inside the root hairs, and the infection thread is formed, and the bacteria enters and infected the cortical cells. Inside the cortical cells, the bacteroids they are formed, which are the polyhedral structures, and a pigment is formed which is leg hemoglobin, and it is a oxygen scavenger. It creates the anaerobic condition, and that is required for the functioning of the nitrogenase enzyme, which is a iron and molybdenum containing pigment, and actually it weakens the nitrogen bond. And here the hydrogen, it is produced, it comes through the reduced enzyme, and first the diamide is formed, then hydrazine is formed, and the ammonia is formed, right? Here you can see that the weakened molecule of nitrogen, it is acted upon by hydrogen from a reduced coenzyme, and the diamide is formed, the hydrazine is formed, and the ammonia is formed. So this is what the story behind the symbiotic nitrogen fixation. You can see here the equation, right? I just want to highlight the important points. This is what the three processes: ammonification, nitrification, denitrification, also the pigment leg hemoglobin, है ना? And these are the chemical attractants from the root hairs to attract the bacteria, and the nodule is formed by the invasion of the bacteria, and the enzyme which is responsible, that is nitrogenase, and we have seen the reaction here. Ammonia, ultimately what is formed? Ammonia is formed. Now the free living nitrogen fixers, it is having the processes like the ammonification, nitrification, and denitrification. For that we have discussed much the different types of bacteria they are involved. For them also we have discussed. Now the nitrate assimilation in plants, where the nitrate first converted into nitrite, and then the nitrite it gets converted into ammonia. And here we have to be clear with the enzymes. Here when the nitrate it gets converted into nitrite, the nitrate reductase function, and when the nitrite it gets converted into ammonia, the nitrite reductase takes part, right? These are the coenzymes, and this is the electron carrier. Now after that, what happened? The ammonia, it is used for the formation of amino acids, because amino acids they join to form the polypeptide, which is considered as protein. So the amino acids they are the constituents of proteins, and here first of all ammonia it gets converted into amino acids, and this is by the two processes. One is reductive amination and second is transamination. Reductive amination, where you can see here that the ammonia reacts with the alpha ketogluteric acid and forms glutamate or glutamic acid, right? The enzyme is glutamate dehydrogenase, and in the second transamination, you will be able to see here it involves the transfer of amino group from one amino acid to the keto group of a keto acid, and the enzyme is amino transferase, where the alpha ketogluteric acid and the aspartic acid it is formed. Now, one more point, that is the formation of amides in the higher plants. Amides, how they are formed? Actually, amides they are formed by, they are the derivatives of amino acids in which the OH component of carboxylic group, right? OH component of carboxylic group, it is replaced by the amine group. So here the amides they are formed by the combination of ammonia and amino acids, and the two main amides they are glutamine and aspergine. I must write here the asparagine, right? And these are the two important amides having the additional advantage for the storage of excess nitrogen and the transport of nitrogen. And as amides, they are having the more nitrogen in comparison, so they are transported through the xylem vessels as well. And the last point is what? Where the urides they are formed. What are urides? Actually, along with the transpiration stream, the nodules of some plants like soya bean, they export the fixed nitrogen as urides. And urides, they are having the high nitrogen to carbon ratio. So this is what the mechanism of nitrogen fixation. I just assume that you will be able to understand the nitrogen cycle and nitrogen fixation, and we will meet in our next video. Till then, have a very good bye.